joined by three men who are at the cutting edge of what is going on around this topic here in St. Louis. Let me introduce the table that we have to start off with here tonight. Dr. John DiPerzio, the Siteman Cancer Center, Dr. Rick Wilson, with the Genome Institute, and Dr. William Galanders, Washington University School of Medicine. Thank you all for being here.
Thank you.
Let me just start by asking you where we are in this fight against cancer. In the documentary, it goes back to President Johnson. Every president from then on has talked about we're on the cusp of a breakthrough in their State of the Union speech. Where are we now? Are things any different now than they were 10 years ago?
I think so. The main thing is that we know a lot more about the disease now than we did before. Knowledge is power. You can't fix anything unless you know what the moving parts are. So that's been the major advance over the past decade. We really understand the process a lot better. Treating these diseases is going to take us into the next decade. It's going to be very difficult to develop treatments that are really effective in these complicated diseases. But it's all possible if you know what causes the disease and what are some of the underlying abnormalities that contribute to the disease.
Is this a disease we can cure, or is this something we will always have and we're learning to treat it differently? How do you approach cancer? What are you thinking about? What are you talking about?
Well, there's an important concept to understand is that cancer is really a whole host of diseases, and you can't treat everyone the same. So while you see specific cancers where there's a tremendous amount of progress having been made in terms of developing new therapies, these aren't effective across all patients. So one of the things that we've done in the last 10 years to understand these diseases is to start to look at their DNA and to understand, what is the defect at the heart of this particular disease, this particular patient's disease, and what can we learn about that to treat it more effectively.
So it's different from cancer of the liver to kidney cancer, or can it be different from two kidney cancer patients even individually?
Yes to all of the above. Lung cancer and kidney cancer can be very different. They can also have similarities, but even two lung cancer patients can have a very different disease that needs a very different treatment.
It's been said as we've been getting ready for this show that you don't want to be too tongue-in-cheek about cancer, but if you're going to have cancer, St. Louis is a good place to have it. Is that true?
Well, I think that there are national leaders in St. Louis in the treatment of cancer, and we are making remarkable strides in understanding cancer. And one of the things that I would just echo that Dr. Wilson said is that we are starting to look at the DNA of cancer. And one of the amazing things that we're finding is that it's incredibly complex. And so even though we've learned so much in the last decade, we've also come to appreciate that it is an incredibly complex disease, and the answers will be difficult as well. So I think that's important to remember.
What are you talking about at the Genome Institute? Because I know you had one of our senators was there recently, along with the head of the National Institute of Health, and you gave a presentation. What's going on with the DNA with cancer in your world?
Well, I think in a lot of ways we're just scratching the surface. I mean, in the laboratory over the last 20 years, we've learned how to do some pretty amazing things. And the thing that we're really talking about a lot these days, as we've already touched on here, is how do we use the patient's DNA, how do we use their genome to understand what's gone wrong? And one of the things that we've done with government funding from the National Cancer Institute over the last 10 years is to start to look at the DNA of several patients, several hundred patients with the same type of cancer to learn what goes wrong in this type of disease. A discovery that our lab actually contributed about 10 years ago was a discovery that some patients with lung cancer, and these tend to be non-smokers with lung cancer, actually have a mutation in a gene that is very treatable by a particular new drug. And so one of the things that happens now for a newly diagnosed lung cancer patient is they want to know, do they have a mutation in this gene, EGFR? And if so, they're likely a very good candidate to go immediately on that targeted therapy.
So is there something to do with the world that we live in terms of our technological advances that allows you to do what you do? Is it all tied together?
It is. I mean, we launched a project back in 1989 called the Human Genome Project, which us biologists refer to as our moonshot. And the idea back in those days was, let's sequence the entire human genome, at least from one person, before we try to do it on additional people, to see what we can learn about our basic operating system, if you will. We started that project long before we had the technology to actually accomplish it, which is exactly the same thing we did when we decided to go to the moon. And through the course of the project, we actually built up technology that allows us now to be able to sequence an individual human genome in just a few days for just about $1,000.
What do you guys talk about when you're amongst your colleagues? What are the other innovations that we'll soon hear about in coming out of St. Louis or research across the country or other parts of the world? What's exciting you about the fight against cancer?
I mean, I think we're, in addition to really being a leader in the world of cancer genomics, I think we're very good at developing basic science insights into some of the aspects of normal physiology that affect cancer growth, like immunology and immunotherapy. How does the immune system actually control diseases on a day-to-day basis?
Getting your body to fight the cancer itself?
Is cancer only a disease of cancerous cells, or is it also a disease of an immune defect such that the cancer gets a little bit of a break from the normal immune surveillance of the patient? And we can focus on that and understand some of the basic issues relating to immunology and take advantage of those understandings to sort of target the immune system to actually make it work better. And if you can make the immune system work better, you're going to actually control diseases much better in general. It's almost a preventative approach, if you will. The other thing is I just play I'm the ultimate optimist as an oncologist, but I also want to, I'd like people to understand that this is a very difficult problem, as Will was saying. These are very complex diseases. You and I have the same number of muscles, and we have the same number of bones, and we have the same number of nerves, but we physiologically probably behave very differently, right? We respond to stimuli differently, we adapt to our environment differently. So there must be differences beyond our genetic code that actually allows us to grow faster or move quicker or think better or adapt to our environment better. So those are the kinds of things that, above and beyond knowing the sequence of a tumor, people have to figure out in the next 5, 10, 15, 20 years. And I think one of the things that I worry about is that, that there has to be a realistic expectation in that, for instance, 50, 60 years ago, a disease called sickle cell anemia is a disease where there's only one nucleotide change from the normal genetic code for one of the genes that encodes for globin gene synthesis for hemoglobin. And when that one genetic mutation occurs, you get this disease called sickle cell anemia, which is a terrible disease. Knowing that single mutation, it's still taken us 50 to 60 years to figure out all about the physiology of that disease, how that one little mutation, never mind a cancer cell that may have hundreds of mutations, how that one little mutation can affect the biology of the blood-forming system, and also how do you fix it? How do you fix it? It's not easy. Suddenly, I think, over the past 10 years, we've been developing some of the
Does that mean we're 50 or 60 years away from?
I think we cure some diseases right now. There are a number of diseases that are curable that were not curable 10, 20 years ago. Childhood leukemia is an excellent example. Chronic myelogenous leukemia in the adult setting. We treat people with a drug that's a targeted drug that puts people into a chronic phase of the disease where they can live a normal life like you and I. And so that they really aren't affected by their disease whatsoever, which in my view is a cure.